Okay. All right. I think let's get started. So yeah, welcome everybody to our CMFI Mass Spec Seminar Series, round 2. It's my great pleasure to have Professor Xiaowen Liu today giving the lecture on top-down proteomics, and I assume he will also talk a little bit about their awesome tool topic his lab is developing. And yeah, to just give you a very brief introduction. So Xiaowen studied computer science at the Shandong University in China. Before he did also a PhD in computer science at the Hong Kong University. And then he moved on to first Canada, to Western Ontario University to do a postdoc, and then also to UCSD in California to work with Pavel Pesner, where I think during that time they developed a first version of today topics, also in the realm of top-down proteomics, which he then I think mainly developed in his own lab at Indiana University and Purdue. And I think now recently he moved on and is a full professor for biomedical informatics now in Tulane University. And yeah, without further ado, thanks so much for giving the lecture today, and I'm very much looking forward to it.
Yeah, thank Daniel for the introduction. I'm very glad to have this opportunity to talk about the research we are doing in our lab. Today, I'm going to talk about proteoform identification and quantification using top-down mass spectrometry. Let's see. Yeah, this is the overview of this talk. So first, we're going to talk about, I think we'll skip the first part, maybe, tandem mass spectrometry. Then first, we'll talk about top-down and bottom-up mass spectrometry. And then next, we're going to talk about how to do proteoform identification and also proteogenomics study of proteomics. And finally, we'll talk about some deep learning methods we just developed for top-down proteomics. Yeah, I think most of you are familiar with tandem mass spectrometry. And I think I'll skip this part. Yeah. So in my lab, we focus on develop tools for top-down mass spectrometry. Right? The main difference between top-down and bottom-up mass spectrometry is that in top-down, we do not do this digestion of proteins, right? Because many proteins have hundreds of amino acids. It's not easy to analyze those proteins directly using a mass spectrometer. So routine bottom-up approaches involves a step of digestion. That is to to digest those digest the protein, long protein into short peptides, then use mass spectrometer to analyze these short peptides. But in top-down mass spectrometry, we skip this step and we analyze intact proteins directly. So that will make the analysis more complex than bottom-up mass spectrometry. Then why do we study top-down mass spectrometry? One reason is to study proteoforms. Proteoforms are products of genes, right? And there are many types of alterations on proteoforms, like mutations and post-translational modifications. Right? So here we have an example. So if you study a protein sequence, you can get a reference protein sequence from a database, right? For example, you can get a protein sequence from UniProt, right? The knowledge base of protein sequences, right? But if you analyze a proteoform in a sample, you can find out the proteoform in the sample may be different from the reference sequence in the database, right? There are many types of alterations. Here, I give an example. The bottom sequence is a Proteoform we are analyzing, and the one above it is a reference sequence in a database. There may be many types of alterations. For example, some N-terminal segments may be removed, and there may be a mutation between this R and K, and maybe insertions and deletions. And also there may be some post-translational modification sites, right? Like it's here, right? This cysteine and serine, they may have PTMs. We can use top-down mass spectrometry to analyze these modified proteoforms. And because of the alterations on proteoforms, we find out that at the proteoform level, the complexity is much higher than the genome level in human cells. Right? Human cells, the number of genes is around 20 to 25,000. But if we look at the proteoform level, the number of proteoforms of a human cell is estimated to be more than 1 million different types of proteoforms. And these proteoforms have their functions. To study those proteoforms, we can use either bottom-up or top-down mass spectrometry. But bottom-up mass spectrometry has its limitations. For example, if you want to study a PTM of a protein, you can use bottom-up approach to do it. But if you, let me see. But in this digestion step, we generate many short peptides, and only a small fraction of these peptides can be identified. So the modification site may not be identified in those peptides, right? So the site may not be ID'd. But if we do top-down approach, so the whole proteoform will be analyzed directly, and the PTM site information can be identified by matching those fragments of this proteoform to the database sequence. Right? And also there are also some other advantages of top-down mass spectrometry, right? One example is to identify this digested proteoforms, right? If you have proteoform, that a protein that is digested, proteolytically digested into 2 fragments, you can use top-down mass spectrometry to directly identify these 2 proteoforms, right? And another application of top-down mass spectrometry is to study proteoforms with multiple modifications, multiple PTMs. For example, in this protein, in these methionine residues, maybe there are some oxidation modifications. Right? If there are 2 oxidation sites, right, and it's not easy to figure out the proteoform pattern using bottom-up mass spectrometry, because the digestion process breaks the protein into short peptides, and we cannot reconstruct the original proteoform with PTMs from these short peptides. If we use top-down approach, right, we can find maybe 2 proteoforms. The first one has modification sites on the first and the third methionine, and the second has a modification site on the second and the fourth, right? Those give us the PTM patterns of proteoforms, right? So here, the reason we want to use top-down mass spectrometry study proteoforms is that it can identify proteoforms with alterations, right? It can also identify these PTM patterns and give us this 100 protein sequence coverage. And then what is the main challenge? The computational challenge in top-down mass spectrometry data analysis. I think the challenge comes from alterations, sometimes most time PTMs in Proteoforms. Right? A common used approach for protein identification or peptide identification in mass spectrometry data analysis is to match a mass spectrum to a theoretical mass spectrum of a database sequence. But when the Proteoform has modifications or alterations, the experimental mass spectrum of this modified form is different from the theoretical spectrum generated from database peptides or protein sequences. Right? Here we give an example. Right? We try to match an experimental spectrum to a database sequence. Here, to simplify the explanation, we only consider prefix fragment masses. For example, if you use HCD, we only consider B ions, right? And suppose we get these B ions for all prefixes. So there are no missing peaks for those B ions, right? Even if we use this kind of ideal case for the experimental spectrum, so it's still not easy to match this experimental spectrum to its corresponding database sequence. When we generate a theoretical spectrum from the database sequence, right, we get a list of peak positions. When we match those peak positions to the theoretical one, we find out from the third peak, right, the peak is shifted to the right. The reason is that the serine residue has a +18 modification. This should be a phosphorylation. Right? So, and also all the peaks right to this third peak, they are all shifted to the right. Right, the shift is around 80 Dalton, and also there's another oxidation on this N residue. So the shift of the peak right to this N would be 80 plus 16, it would be 96. Right? So you need to consider all these different types of modifications and shifts, sometimes a combination of two shifts, right, to match your experimental spectrum to the database sequence, right, to the theoretical spectrum generated from database sequence. Right? And another challenge in top-down mass spectrometry data analysis is that some proteoforms, or sometimes they're similar protein sequences, they are highly similar to each other. Maybe you have only one or several mutations if you compare two sequences. How can you map a spectrum to one of these similar sequences confidently? That is another challenge. Another computational challenge is to study these highly modified proteoforms, like histone proteins. Here we give an example of this histone H4 protein. So these red triangles and this annotated this colored positions, they are possible modification sites. So there are more than 10 possible modification sites, and for each site, there are several possible PTMs. So the number of combinations of these possible size can be huge, right? Can be maybe millions or billions of forms. Then when we have a mass spectrum of this protein form, how do we match this protein form or match this spectrum to the modified protein form of this protein, right? At the bottom, we give an example, right? This is an identified protein form of histone H4, right? It has 5 modification sites, right? We annotated with the red, these circles, right? The acetylation or ACDM dimethylation, right? But many of these protein forms are unknown, right? They are not stored in databases. How can you identify these modification sites directly using top-down mass spectrometry? Yeah, here we listed some challenges, right? First, they have pre-processing is not easy, and also it has multiple modifications, right? And how do you do database search in a fast speed, and how do you distinguish between positive and negative IDs? Right? Those are all computational challenges. Right? So next, when we do top-down mass spectrometry data analysis, we have mainly 2 steps. One is a pre-processing, we call it spectral deconvolution. We convert this top-down mass spectra to a mass list, right? This to simplify the data. Then we search this mass list against a protein sequence database to ID, right? To identify a protein form. Some of the forms have PTMs or other alterations. Okay. Then the next part is how do we identify protein forms? Right? As we discussed, the most commonly used approach is to do database search. Right? That is to search an experimental top-down spectrum against a protein or protein form database. And for each protein form sequence in the database, we'll generate a theoretical spectrum. Then match this theoretical spectrum and the experimental one. Right? If we find a match with a high similarity score, then we can report it as an ID. And the challenge is to identify PTMs, right, or mass shifts in these protein forms. To solve this problem, I think about 10 years ago, we developed a tool called MS-Align, right? That is to use spectral alignment to do this kind of match between mass spectra of modified protein forms and database sequences. Right? So the idea is we list all these peak positions of the experimental spectrum on this in this 2-dimensional table. Right? Each row here corresponds to a position, peak position of the experimental spectrum, and for each column is corresponds to a position of the theoretical peak position of a protein sequence in the database. If there's no mass shift, no PTM, we can find these diagonal lines with these intersections, like this black diagonal line and this blue diagonal line. And if there's a PTM, then our shift will have this vertical, this shift, and the length of this red line here equals to the mass shift of the PTM. So using this approach, we can match a spectrum of a modified protein form with a database sequence without modification. And we can also specify how many mass shift we want to identify in this match. And finally, we can report a similarity score. that is the number of matched these intersections in this path. So after we identify this spectrum, the next step is to assign a confidence score to this modified, to this mass shift. How do we do this? We use a Bayesian approach. That is, we try to enumerate all possible modified forms for a mass shift. For example, for this mass shift around 42, we'll look at all possible these acetylation sites. Right? And for each site, we'll generate a theoretical spectrum and compare the theoretical spectrum against the experimental one and get a similarity score. Based on the similarity score, we can find the site with the best similarity score and also its confidence score. So the challenge here is that the number of candidate these proteoforms with these modification sites can be large. There can be thousands of these forms. So we need to do the calculation of these similarity scores quickly. Right? And so we can use a dynamic programming approach to do it. The next challenge, as we talked, is to identify proteoforms with multiple PTM sites. Right? So when a proteoform has a variable PTM, say phosphorylation, right? So it's possible that the sequence has maybe tens of possible phosphorylation sites. If you try to enumerate all modified forms of this protein, you can find out there's this combinatorial explosion problem, right? You can get many proteoforms, right? Candidate proteoforms generated from these potential sites. That will make your computational time very slow, right? So how do we solve the problem, right? So here we also show the previous example, right? For example, if you want to identify a modified proteoform of histone H4, there are so many possible modification sites, and the different types of PTM types can be added to those modification sites. Right? To solve this problem, we first formulate is also a database search problem. And for each sequence in the database, so the goal is to find a modified proteoform that can be matched to the experimental spectrum with the highest score. Right? Here we have a reference sequence, and also we have a set of variable PTMs. Right? And we want to look at all possible modified forms of the reference sequence and find the best one. Right? To solve the problem, we first need to represent these modified forms. Right? You have a sequence, a reference sequence, and you have a set of variable PTMs. Then how do you represent all possible forms generated from this reference sequence and this set of variable PTMs? Right? Our solution is to use a graph to represent all possible forms. Right? So given a sequence, a reference sequence like this ARK, TDDR here, we use a node to represent a peptide bond in the sequence. Right? For example, this X1 represents the bond between A and R. Right? X2, the bond between R and K. Then we use edges to connect these nodes, and there may be multiple edges connecting two nodes. And each edge represents a residue. Sometimes it's unmodified, sometimes it's unmodified, sometimes they are modified. Right? For example, this R residue, there are two possible modification types, right? Acetylation and methylation. Then we'll have three edges. One is unmodified, one is with acetylation, and the other is methylation. Right? Then we can generate this graph, and after this graph is built, each node, each form of this protein sequence with these variable PTMs is represented by a path in this graph. Right? So for example, this bolded, this red path just represent a modified protein form. So, and all possible forms can be represented by this graph. Right? So that means this graph contains the complete information of your input. Similarly, we can use a graph, not graph, maybe a path to represent an experimental spectrum. Right? So each node here is a peak, and the edge connecting two neighboring peaks is annotated by the distance between these two peaks. So this approach, we can match those, we can use a simple graph to represent all protein forms. And that is better than the traditional graph approach, in which the amino acids are represented by nodes. If you represent amino acids by nodes, you'll find out you have many additional edges to represent the combinatorial patterns of PTMs. So then after we build this graph to match a mass spectrum to a modified protein form, the problem is converted to another problem. That is, you want to find the two paths in the graph so that these two paths have shared prefixes with. So I'll show how to show a result. Right? For example, here we can find this red, this bold path, right? This 198, 142, and 181, and 115, and 71. That is the path in the graph. And this one can be matched to the spectrum, right? This Y0 to Y2, the distance is 198. And Y2 to Y4, the distance is 142, right? Then you can find out this, and also Y6 to Y7, the distance is 186. That is the sum of 115 and 71, right? So you can match these two paths together and to get a pair of paths. That is the identified protein form from the graph. So this can also be done by using a dynamic programming approach. So the next question is, after we identify these modified forms, how do we assign a p-value or a confidence score for the ID? So for this protein form IDs with multiple modifications, we use a method called MCMC, Markov chain Monte Carlo, to simulate some possible modified forms. And then using these simulated samples, we can estimate the distribution of the similarity score between a random Proteoform and the experimental spectrum. Based on this distribution, we can compute a p-value. And here we also use an approach called importance sampling. So that is when the score is high, we'll increase the number of sampling that are similar to the modified Proteoform. This approach can reduce the number of samples in this sampling approach. Next, we use this method to study two cancer cell lines. So this is a collaboration with Dr. Liang Sun at Michigan State University and Dr. Amanda Hammond from Ohio State University. Right, we compared the two cancer cell lines. One is SW480. That is a non-metastatic cancer cell line. The other is SW420, right? This metastatic, this colorectal cancer cell line. Now, so Dr. Sun used different approaches for proteoform separation, like this CZE and RPLC. We also use approaches to combine several separation methods. Using these different approaches, we could identify more than 17,000 proteoforms from the SW480, right, or around 14,000 proteoforms from the other cell line. Which is about a threefold increase compared with the best existing these cell line studies. And we identified many PTMs, right? The most common ones are N-terminal acetylation, right? There are more than 4,000 forms with N-terminal acetylation sites, and also phosphorylation, methylation, and oxidation. Those are common PTMs, right? So here is an example, right? This is PDAP1, that is a cancer prognostic marker. That is, so in this protein, we identified a mass shift around 160 here. So that's corresponding to 2 phosphorylation sites. And also we did this differential expression analysis. We compared the abundances of proteoforms in these two cell lines and identified hundreds, more than 400 differentially expressed proteoforms. Right? If we do this clustering analysis, we generate triplicate, right, of SW420 and a triplicate of SW4, triplicate from SW620 and triplicate from SW480. So you can find out that using these differentially expressed proteoforms, we can easily classify the samples into 2 groups, right? And for these differently expressed proteoforms, we also look at some special cases. That is, we look at 2 forms from the same protein, and we found out that for some proteins, they have 1 proteoforms that are overexpressed in SW480 and another proteoform that is underexpressed in the same cell line. So that is, if you look at the protein level, you cannot find those differentially expressed proteins. But if you look at the proteoform level, you can find these differently expressed proteoforms, right? Some are overexpressed, some are underexpressed, right? For example, for this DAP protein, we identified two forms that are either that one is overexpressed, the other is underexpressed. And these differentially expressed proteins are also related to these functions, cancer-related functions, right? Like growth of tumor, right? After we do this pathway analysis of these proteins. So next, we also did a proteogenomic analysis of proteoforms of these cancer cells. Right? The idea is to use RNA-seq to identify mutations in those cancer cell lines, then build a customized protein database. And then we can use our database search tool to identify these proteoforms with mutations. To build a database, we first find non-synonymous SNPs, right, from RNA-seq data. And these SNPs can be divided into two groups, homozygous and heterozygous. Right? For heterozygous, we'll consider the combinations of these mutations and build their corresponding protein sequences. So from one database protein sequence, we may generate several protein sequences with mutations. Then we use this customized database for protein identification, for proteoform identification. So we identified 111 proteoforms with single amino acid mutations from 82 genes. Right? Some of the mutations are on these cancer-related genes, like TP53, that's a tumor suppressor. Right? And we identified a mutation on this R residue. Right? Another example is this MSH6. So we also identified a mutation on this E residue. Right? Those mutations are identified in SW480 cells only, not in the other cell line. So these mutations and maybe this cell line specific. Right? Okay. So finally, we'll briefly talk about some deep learning methods we developed in my lab. So we started to do deep learning like 5 years ago. So we are still new in this area. So the first part is to do envelope evaluation. That is a function in spectral deconvolution. Right? After you identify a isotopic envelope in a top-down mass spectrum, how do you assign a score to this identified isotopic envelope? So we tried to use a machine learning approach to design a scoring function. Right? The idea is we use the isotope distribution to get a theoretical isotopic isotopic peak intensity distribution of a proteoform, like the red one. Then we match it to a experimental isotopic envelope in the spectrum. Then we can get a matched envelope pair. Then the envelope pair will be the input of a CNN model, this convolutional neural network model, to get a score. Right? So we converted a matched matched envelopes or matched pair of envelopes to a matrix, right? So in this matrix, we have scores like the peak intensity and the differences between matched m over the m over the errors of matched experimental and theoretical peaks, right? And also the difference between the theoretical peak intensities and the experimental peak intensities for this kind of information, right? Then we also built a training data set, right, for this problem. So we label all those matched B and Y ions as positive ones, and also some these envelopes that cannot match to any theoretical ions will be labeled negative, right? Using this approach, we can train this convolutional neural network model. Right? It has several blocks. In each block, we have 2 convolutional layers. And finally, we are connected to fully connected layers to do prediction. So the output will be a confidence score between 0 and 1. Using this approach, we can increase the prediction. We can increase the ability of this scoring function to distinguish between positive and negative samples, negative data points, right? So here is a comparison of this ROC curve between this neural network model and our previous model. Is the green one is MSDConv, is our previous model, and the orange one is the neural network model, right? So on these 2 data sets, we can see the neural network model is much better than the previous MSDConv scoring function. Right? Another problem we studied is to predict the retention time of proteoforms in top-down mass spectrometry, or the migration time of in this capillary zone electrophoresis separation, or CZE separation. Because there are some existing models for bottom-up retention time prediction. So we just borrow some models from bottom-up mass spec retention time prediction and apply them to top-down mass spec data analysis. The idea is to first we just use protein sequences, right, and use different machine learning models to predict their retention times or migration times. Yeah, we test the different type of models, like fully connected neural network, this recurrent neural network, and convolutional neural networks. So the data set was the training data set was generated from ovarian cancer top-down mass spec data. So that is from Sante also. Sante's Nature method paper. Right? So in this data set, they generated 10 replicates of the same sample. So we also generated 2 training data sets. One is from the first replicate. The second is to combine all this data from 10 replicates. We call them LC1 and LC10. Right? And also we collect some bottom-up data. We call them LC peptide. So the bottom-up data was generated for transfer learning because the top-down data is pretty small. To train such a complex model, we wanted to combine bottom-up and top-down data to increase the samples, the training data set size. If we train the models on the top-down data directly, so the performance is not very good, right? So if you look at this GRU, this is the Prosit model, right? It's similar to the Prosit model. Its performance is similar to some very simple linear regression models or random forest regression models. But if we combine top-down and bottom-up together, right, we find out that the accuracy, right, the R value and the mean absolute error, and also this delta, it's called TR 95%. That is the width of the area that contains at least 98.5% of this predicted times. So when we combine top-down and bottom-up, we find out the performance is much better than using top-down data only. The idea is to use bottom-up to train the model first. Then the pre-trained model will be retrained using top-down data. And that can increase the performance of the deep learning models. So that is for LC retention time prediction. For this migration time prediction in CZE data, even if we do not do transfer learning, the performance is pretty good. So the reason might be that this migration time prediction is simpler than this retention time prediction. So because it's determined by the charge state of the Proteoform and also the mass. So we try to use this retention time information to help us to remove this incorrect IDs in database search, right? So that's one experiment we did, right? So we get the histogram of target IDs and decoy IDs for each range of these E values, right? So the left ones have high E values, right? You can see here, for this E value range, the target is 57 and the decoys is 8. After we do filtering using this retention time prediction, the decoy is reduced to 3 and target also is reduced to 53. But we can see that most of the target are still reported, right? For this other example, right, the decoy is 8 and the target is 61. After we do filtering, the decoy is reduced to 1. Right? The target is also reduced to 52, but we still keep most of the target IDs and removed most of the decoy IDs. It just shows that this prediction can be used to increase Proteoform IDs. Yeah. Finally, we are maintaining this topic suite software tool. If you are interested, you can download it from this website. And these are my collaborators. Yeah. So thank our collaborators for helping us to improve the software tool and also to provide us with many new problems and new data sets for our research. Yeah, I think that. Thank you all. Any questions?
Right. Thanks so much, Xiaowen, for this very interesting talk and insights in your new developments. This is very exciting. Yeah, I think we will have a couple of minutes for questions. But yeah, before we move on to the questions, I just wanted to thank you again for presenting, and yeah, everybody for joining today. And if you don't stay on for the questions now, I hope to see you next time.